Hi, everyone. Thanks for joining today. We're going to give it about one more minute for folks to get dialed in. All right. Hi, everyone. Thank you for being here today. I'm Connie Mitchell. I'm a scientific program manager at the Health and Environmental Sciences Institute, or HESI. And today we have a webinar as part of our Next Generation Ecological Risk Assessment Committee. So just a little bit of housekeeping and background before we start today. So here is the mission of our committee. So it's to develop, refine, and communicate scientific tools and approaches needed to support ecological risk assessment around the globe with a focus on alternative non-animal testing methods. And some of our sub-goals in this are to use and development of alternative methods, and also to come up with tools to evaluate toxicokinetics for organic chemicals. So just a little bit of logistics today. So for this webinar, it will be recorded. The recording will be made available to the registrants and it'll be put on our HESI website. Participants other than the speakers are muted. So questions should be typed into the question box in the control panel. I'll have a slide on that on the next one. And if you have any issues during the webinar, you can feel free to email me. So it's cmitchell@hesiglobal.org. So there's just how to ask a question. If you're on the desktop version, you should be able to see it on the sidebar where you can ask a question. And then on the web instant join, there's a question mark you should be able to select. Just a tiny bit about our current portfolio for the Ecological Risk Assessment Committee. So as shown before, we have topics related to toxicokinetics and bioaccumulation shown at the bottom for the different fish, invertebrate, and birds. And then we have quite a few projects related to new approach methods. So we have an EnviroTox database and tools that's available for free online to do read across and come up with safe levels for chemicals that are potentially data poor. We have a group looking at new approach methods for endocrine testing tools. We have an effluent assessment group, and then we also have an alternatives to chronic fish testing group, which the work presented today most closely relates to that group, excuse me. And there's just some contact information if you're interested in getting more information or finding out about any of those projects. And with further ado, I'm very excited to have our speaker today. Today we have Dr. Marlo Jeffries giving a webinar entitled Investigating the Predictive Power of Sublethal Endpoints for Fish Embryo Toxicity Test. Dr. Jeffries is an environmental toxicologist and physiologist. She's an associate professor and department chair at TCU. Her research focuses on a few areas related to ecotoxicology, including the effects of contaminants of emerging concern on different processes, including reproduction, development, and immunity. Her lab also works on the development of fathead minnows as a model organism for immunotox testing, and the effects of endocrine disrupting compounds on thyroid hormone signaling and other endpoints, in addition to the development of alternative models for routine tox testing. So thanks so much, Marlo, for giving this talk today. I'm going to give you presenting rights and turn over to you.
Okay. All right, can you see my screen?
Yes, looks great.
Okay, excellent. So thanks to everybody for being here today, and thanks to HESI for allowing me to give a webinar today. I'm excited to talk about some of the research that I've done over the last 11 to 12 years So today I'm going to talk about some of our work related to the Fathead minnow fish embryo toxicity test. And specifically what I'm going to focus on is efforts that my lab has taken to identify sublethal endpoints that may actually be useful as routine endpoints for the Fathead minnow fish embryo toxicity or FET test. I was telling Connie as we were chatting just before the webinar started that I actually really had kind of a lot of fun putting this webinar together because it really gave me a chance to sort of reflect on how the research got to this question of sublethal endpoints. So I actually want to start. Uh-oh, my slides are in here. Oh, there we go. So I actually want to start back in 2011, 2012, when I was a postdoctoral fellow in Jim Orris's lab at Miami University. So while I was a postdoc with Jim, he was actively involved with HESI, and he had actually received a grant from HESI to do some animal alternatives work. And specifically, one of the objectives of the grant that he received was to take a look at the Fathead minnow embryo toxicity test or FET test and compare its performance to the Fathead minnow larval growth and survival test, which is a standardized toxicity testing method used here in the US primarily for effluent evaluations. And at the time, Jim actually had a master's student who was supposed to work on this project. Unfortunately, or maybe fortunately for me, that master's student got really, really homesick during their first semester of graduate school and ended up bailing, just up and left the university. And here we had this, Jim had this project sort of just hanging in the balance. He needed somebody to work on it. So he asked me if I would do it. At the time, my interests were really sort of focused on endocrine disruption and immunotoxicity. I had honestly never really thought much about fish embryos before. But really, I think this sort of strange series of events I really consider to be one of the most serendipitous moments of my scientific career. Because it gave me the opportunity to really expand my research repertoire. And now I can tell you, I think about fish embryos all the time. And this has become actually a major part of my academic life and my scholarship. So when I was working with Jim, obviously we did these comparisons of the FET test and the larval growth and survival test as outlined in the grant. But one of the things that I was able to do when I was working with Jim was actually expand that project a little bit and begin to look at the identification of sublethal metrics for use as additional FET test endpoints. So I do want to talk just a little bit about how we actually got to that idea of wanting to identify such metrics. Before I dive in too much though, I do well, I should mention that this work I've been lucky enough to actually take to TCU with me. So now I have a group of really fantastic students that work on questions like these. And we've been actually really lucky to get funding to help support this work as well. So to start, I do just want to briefly talk about the Fathead minnow fish embryo toxicity test and the larval growth and survival test. I'm sure that many of you are familiar with both of these tests, but I at least want to touch on it in case there's anybody that is less familiar with these tests. So the Fathead minnow larval growth and survival test, like I mentioned, is a standardized EPA test method predominantly used in effluent screening. Excuse me. And in these larval growth and survival tests, essentially what we do is we take newly hatched fathead minnow larvae and we expose them to 5 or more concentrations of a chemical or effluent. And then of course we have a control group. This is a 7-day exposure, and over the course of that 7 days, we monitor mortality of the larvae. And then at the end of the test, the growth of the larvae is monitored. And the metric that we usually use for growth in this case is mass. In the Fathead minnow fish embryo toxicity test or FET test, essentially what we do here is we take embryonic fathead minnows that are at the 32 cell stage or less, and we expose them to 5 or more concentrations of chemical or effluent. And in this test, we do usually have both a negative control and a positive control. The negative control is usually unexposed or potentially exposed to just a solvent. And then we do have a positive control that usually is exposed to something like 3,4-dichloroaniline. In the FET test, mortality is monitored every day, and in fathead minnows, this is a 5-day test. So mortality is monitored every day, and I apologize about the barking dog if you can hear it. Mortality is monitored every day for 5 days. And then at the end of both of these tests, we are able to use the survival data to estimate acute toxicity. One major difference between these tests, aside from the fact that one uses larvae and one uses embryos, is the fact that the larval growth and survival test does include growth as a metric, and that growth metric is used to estimate chronic toxicity. In the larval growth and survival test, or excuse me, in the FET test, we do not have an analogous endpoint. So the FET test is really restricted to estimating acute toxicity and really doesn't have any endpoints currently associated with that test that are able to estimate chronic toxicity. So before I talk about the identification of sublethal endpoints, I think it's important to understand why we became interested in looking at sublethal endpoints. To do that, I just want to show some FET test and LGS test comparison data. So what you are seeing on the screen here are the results of 5 tests, five tests with 5 different chemicals in which we were comparing the FET test to the LGS test. On each of these images, you will see a red line, and that red line depicts the LC50 values derived from the FET test. The purple line depicts the LC50 values derived from the larval growth and survival test. On each one of these images, the diamonds represent the mean LC50 value estimated for multiple replicates of each test type. And then the lines extending from the diamonds represent the 95% confidence interval of that LC50. In cases where the LC50, the 95% confidence interval of the LC50s overlap between the 2 test types, we would say that the tests are equivalent in performance. So for these 5 chemicals, you'll notice that all 5 of them, we see this overlap between the 95% confidence interval. And therefore, for these 5 chemicals, we concluded that these test types have equivalent performance. While this is great, this isn't always the case. We do have cases where the FET test and the LGS test have different outcomes. So they produce different LC50 values. Up here, I've got an example of 3 of those cases with 3 different test chemicals, 3,4-dichloroaniline, DCA, a mock municipal effluent, and then cadmium. And what you'll notice in these cases is that the LC50 derived from the FET test is higher than the LC50s derived from the LGS test. What this tells us is that for these 3 test chemicals shown here, that the FET test was less sensitive than the LGS test. I should note that this finding is not unique to my lab. There are lots of instances in the literature where we actually do see that the FET test is not quite as sensitive as larval tests, juvenile tests, or adult tests. There are some chemicals that this tends to be more common for. So for example, if we have chemicals with large molecular weights, some of those chemicals may not be able to cross the chorion. And if they can't cross the chorion, we're not going to have the embryo exposed to much of that chemical. So that's going to make the embryo less sensitive. We also see this pattern for several neurotoxicants where the embryos just seem to be less sensitive to neurotoxicants than the larvae. So this trend that we're seeing here is not terribly uncommon, and it has definitely been noted in the literature. And this difference that we're seeing here really made me start to wonder, could we identify and then include additional metrics as FET test endpoints that would actually improve the sensitivity of the FET test. Excuse me. So we've actually looked at a variety of potential FET test endpoints. We've looked at spontaneous contraction of the embryos. We've looked at heart rate. We have looked at optomotor response. We've even looked at the expression of genes associated with growth and development. I think if you can name it, we have probably at least thought about measuring it if we have not already measured it. Today, what I want to do is focus on 3 endpoints that we think offer the most promise as additional FET test endpoints. The first of those that I'm going to talk about is growth. And what you see on the slide here is the results of a FET test conducted with 3,4-dichloroaniline. So on the x-axis here, you can see concentrations of 3,4-DCA, and then on the y-axis, you can see snout-vent length measured at the end of the FET test, which in fathead minnows is in about 5 days post-fertilization. And what you'll notice here is that as we have increasing concentrations of DCA, we see decreases in snout-vent length. So this tells us that DCA is somehow inhibiting the growth and development of the eleutheroembryos and embryos that are in these FET tests. To just give you a visual depiction of what this might look like, here I have an image of a control larvae and then or a control eleutheroembryo and a eleutheroembryo that was exposed to 8 milligrams of DCA per liter. And you can see that their length is different, but that's really not the only difference that you can visualize, and we'll sort of come back to that a little later in the talk. One of the things that's interesting about growth is that we actually observe alterations in growth in response to about 70% of the chemicals that we've tested in my lab or when I was a postdoc in Jim's lab. And so this is a relatively common abnormality that we do observe. The other thing to know about growth is it's pretty easy to measure, which offers some real advantages when you're thinking about additional test endpoints. The next endpoint that I want to mention is eye size. And on this image, what you're seeing is eye size at the end of a FET test conducted with 3,4-DCA. Here, what you're seeing is that as we increase the concentration of DCA, we see significant and dose-dependent decreases in eye size. One of the things that I should mention here is that in my lab, when we are measuring eye size, we measure it as eye area. So we actually use image J to calculate the area of one of the two eyes. To give you a visual depiction of what this may look like, I've provided the images of 2 eleutheroembryos up here. In this case, we've got a control embryo and one exposed to 8 milligrams of DCA per liter. And you can see there is definitely a difference in eye size. But again, it's not the only difference that you're seeing in that eleutheroembryo. Here, with eye size, we actually see alterations in eye size in about 75% of the chemicals that we tested. So this seems to be a relatively common abnormality that we observe. The next endpoint I want to talk about is pericardial edema. And again, these are the results of a FET test conducted with 3,4-DCA. And what you'll notice here is as we have increasing concentrations of 3,4-DCA, we see a very nice dose response curve in which we see increases in the severity of pericardial edema. And in my lab, to characterize edema, we're actually looking at pericardial area. Some people will just look at pericardial edema as presence or absence, but we actually are taking the time to measure pericardial area in an effort to sort of get at the severity of that edema. Here are some images provided just to show you what this may look like. Clearly, you can see the pericardial edema in the fish that was exposed to 8 milligrams per liter of DCA. And you can even see that there's definitely some heart malformations happening as well in this eleutheroembryo. One of the things that's interesting about pericardial edema is that we have seen this in response to 100% of the chemicals that we have tested in my lab, and this has been noted frequently throughout the literature. So this does seem to be a common abnormality, and like growth, like eye size, it's also easily measurable and easily observable. So here we have these eleutheroembryos at the end of a FET test that clearly display some developmental abnormalities. But what we don't know is what is the fate of these abnormal embryos. So we have these control embryos these control eleutheroembryos that look really normal, and then we have these exposed eleutheroembryos that have all sorts of malformations and developmental abnormalities. And I remember it was a former student of mine, Kyle Rausch. He gave a presentation at a CTAC meeting. Oh, this was probably seven years ago. And after his presentation, I remember there were a group of us standing around talking about these developmental abnormalities and discussing what the fate of these embryos or eleutheroembryos with these abnormalities might be. And I distinctly remember that there were some people in the conversation saying, oh, these guys are dead. There were others in the conversation saying, they're probably going to be fine. They'll grow out of the edema. They'll catch up in terms of length. You know, their eyes will recover. Everything will be fine. So there really seemed to be sort of two schools of thought regarding these abnormal eleutheroembryos. I personally am looking at an eleutheroembryo like that shown on the bottom left-hand corner of the screen here, thinking there is no way that this fish is going to make it. But we didn't actually have data to support that, right? We were sort of all just using best judgment and sort of anecdotal information to try to predict the fate of these abnormal eleutheroembryos. So this got us really interested in actually generating data to determine what was going to happen to these guys. And this is where my former PhD student, Julie Schiffa, really comes in. A lot of the work that I'm going to talk about today, actually all of the work that I'm going to talk about from here on out, was part of Julie's dissertation research. And Julie, like me, became really interested in this question about what happens to these abnormal embryos. So Julie and I, of course, knew that chemical exposures led to these developmental abnormalities, but what we didn't know is whether or not those developmental abnormalities could cause later life adverse effects. And we were particularly interested in linking developmental abnormalities to later life adverse effects that are relevant for populations. So could we link these abnormalities to adverse effects that have some sort of ecological relevance? So what I'm going to talk about for the remainder of my time today is our efforts to link these abnormalities to these ecologically relevant adverse outcomes. So first I want to talk about the developmental abnormalities that we decided to focus our attention on. And we decided to focus on eye size, snout-vent length, and pericardial area. So these are the same three endpoints that I showed you data for just a few slides ago. And the reason that we wanted to focus on these in particular is one, that they seem to be common, right? So remember that pericardial area, we have seen development of pericardial area for 100% of the test chemicals that we've looked at. And then for eye size and snout-vent length, also seeing a really high incidence of reductions in eye size and reductions in snout-vent length. Another reason that we were interested in these three in particular is because they're easy to measure. So we can simply measure these by taking a photo of the eleutheroembryo or embryo and then using image J to measure these metrics. So really simple and easily observable in terms of measurement. Another reason that we wanted to focus on these is we do have some evidence that reductions in eye size are related to neurological development. And one of the things that I mentioned earlier is that the FET test seems to be less sensitive for neurotoxicants. So we thought, okay, well, if eye size is a good predictor for neurological disruption, well, maybe that provides us an additional endpoint that can really be of use when we are dealing with a neurotoxicant. And in the case of pericardial area, one of the things that we can assume about pericardial area, especially in severe cases where it actually coincides with cardiac malformations, is that this is going to be linked to cardiovascular function. So these are the three endpoints that we decided to focus on. And then we also had to pick the ecologically relevant adverse outcomes that we wanted to take a look at. And we actually looked at 5. The first one that we looked at was feeding ability. So in this case, we were interested if our eleutheroembryos, after they had grown up a little bit, could actually chase and capture prey. And of course, this is an ecologically relevant behavior because if you can't feed, you are probably not going to survive for very long. We also looked at C-start response. C-start response is a instinctual behavioral response to predators. And in this response, basically what larval fish do is if they detect vibrations in the water or other stimuli in the water that are associated with the predator, they will initiate an escape response. And that escape response involves them sort of twisting their body into a sea-like shape and darting away from the perceived predator or the perceived danger. So in this case, that C-start response is ecologically relevant because it is linked to predator avoidance behavior. If you can't avoid predators, you're going to get eaten, and that you're not going to be able to contribute to your population any longer. Another thing that we looked at was swim performance. We're dealing with fish here, so obviously the ability of a fish to swim is going to be pretty important for a variety of reasons. It's going to contribute to the ability to feed, it's going to contribute to the ability to escape predators, to find mates, to move away from other dangers. So swim performance is obviously going to be pretty ecologically relevant for fish. Another thing that we looked at was growth performance. So growth is going to be important from an ecologically or from a population level standpoint, because if an individual cannot grow and they can't reach sexual maturity at an appropriate time, they may not be able to reproduce during the typical mating season, which is obviously going to have some population level consequences. And then the last thing that we looked at was longer term survival. So all eleutheroembryos that have pericardial edema at the end of a FET test, are they actually able to recover from that edema, and can they survive all the way through larval development? Obviously, if a fish doesn't survive, they're not going to be able to contribute to the population. So again, this would be considered an ecologically relevant outcome. So in order to link these developmental abnormalities to these ecologically relevant outcomes, Julie set up a really nice experimental design. And essentially what she did is she conducted standard FET tests using fathead minnow embryos at 32 cell stages or less. And then the FET tests ended at 5 days post-fertilization. So these are just normal FET tests that she was conducting, and I'll talk about the chemicals that she used just here in a moment. And then at the end of that 5-day FET test, she actually took photographs of all of these surviving eleutheroembryos. After taking photographs, she transferred the eleutheroembryos into individual beakers containing clean water. So at this point, she has removed them from their exposure solution, and she then raised them to 14 days post-fertilization. So she had 9 days from the end of the FET test to the end of her sort of experimental period. And during that 9-day period, she looked at a variety of endpoints. So at 8 days post-fertilization, she conducted a feeding assay to determine feeding ability of the fish. At 12 days post-fertilization, she conducted the C-start assay to get a sense of predator avoidance behaviors. And then at 14 days post-fertilization, she conducted a swim performance assay to assess swimming ability, and then we also assessed growth in those individuals. It's also important to mention that during that 9-day period in clean water, we were obviously monitoring mortality every day as well. Julie conducted these tests with 3 chemicals: cadmium, 3,4-DCA, that reference toxicant that I've mentioned quite a few times now, and then PFOS. A lot of the data that I'm going to show you today is specific to cadmium. I really don't have enough time to show all of the data. So for the sake of saving time, I'm going to focus primarily on the cadmium data. So first, let's take a look at the results of eye size when our eleutheroembryos or our embryos were exposed to cadmium. So what you're seeing on this figure is cadmium on the x-axis, and what you'll notice is that as concentrations of cadmium increase, we see a reduction in the relative eye size of the eleutheroembryos measured at the end of the FET test. Just to give you a sense of what this may look like, I provided images of some of the eleutheroembryos from these tests on the right. And you can see the eleutheroembryo in the top image has a much smaller eye than the eleutheroembryo on the bottom. So here we have shown that cadmium does induce decreases in eye size. And next, we wanted to see if there were other abnormalities associated with cadmium. So do we see alterations in any of the ecologically relevant endpoints that we selected? Here, I'm just going to talk about feeding ability. So I'm not going to show I'm not going to show all of the data because, for example, we didn't really see anything with C-start for any of the chemicals, so you're not actually going to see any C-start data. But here with the cadmium exposure, we did see that fish exposed to cadmium experienced reductions in feeding ability. So what you're noticing here is really our eleutheroembryos and embryos that were exposed to 40 or 80 micrograms per liter of cadmium really just could not capture prey as well as our control eleutheroembryos. So now Julie's shown that we do have reductions in eye size, we have reductions in feeding ability, but do those things correlate to one another? Is that reduction of eye size predictive of reductions in feeding ability? So to determine that, we did a regression analysis. And what you see here is the results of that regression analysis. What you are noticing here is that we do have a significant correlation, but if you look at the R-squared here, it is not a strong relationship. So in this case, we really concluded that you cannot use relative eye size as a predictor for feeding ability. The next thing we wanted to look at was delayed mortality. I will say in this case, the delay in mortality data, or the data that's shown here, was actually derived using data from the cadmium exposures, the PFOS exposures, and the DCA exposure. So this is for all of our chemicals. And essentially what we were trying to do here is determine, is there a reduction in eye size? Is there a sort of severity of that reduction that allows us to confidently say that that individual is going to die? So what we did is we took data from all three of our chemical exposures and we did an inverse prediction model to determine how bad the reduction in eye size needed to be to get to either 80 or 95% mortality. So what you see here in the yellow is basically showing that in order to achieve an 80% mortality rate in the population, relative eye size would need to be reduced to 0.41. If you wanted to achieve a 95% reduction in mortality, eye size would need to be reduced to 0.01. In the case of eye size, we did not have any instances where the relative eye size of the eleutheroembryos at the end of the FET test was below 0.5. Essentially what this means is that eye size can't be a good predictor of delayed mortality because we never actually see eye size reductions severe enough to achieve significant levels of mortality. The next endpoint I want to talk about is length. And here what you're seeing is our data from our cadmium FET tests. And you'll notice that as cadmium concentrations increase, we do see significant decreases in the snout-vent length of the eleutheroembryos at the end of the FET test. And again, here on the right, I've shown just some images to show you what those differences in length may look like. But again, as you're seeing hopefully a repeating pattern here, that it's never really just one malformation that we see. There's clearly lots of other things going on with that eleutheroembryo in the top image. So we do see reductions in length in response to cadmium. And then of course, Julie wanted to measure all of her ecologically relevant endpoints. And the one that I'm going to talk about here is growth performance. Excuse me. So with regard to growth performance, basically what Julie saw is that when she measured the size or the length of the fish at the end of 14 days, so after they had been transferred to water and grown out, we did see some reductions in length. However, these were not significant. And one of the things that you will notice here is in this 80 milligram per liter group, we do have quite a reduction in length, but our sample size for that group was only 1 because we only had 1 survivor. So while we don't have statistical significance here, it sure does look like cadmium is reducing the length of the fish, particularly in that highest dose. When we tried to correlate length of the eleutheroembryos at the end of the FET test with the length of the embryos at the end of that grow out period, we did see a significant relationship between the two. However, our R-square again was pretty weak, only about 0.29. So we don't necessarily have the strongest relationship here. So we weren't comfortable concluding that length measured at the end of the FET test is a good predictor for growth potential or growth performance over the long term. However, when we took a look at delayed mortality, we did see that delayed mortality could be predicted by length. Again, for this inverse prediction, we are using data from cadmium exposures, DCA, and PFOS exposures. So this is data from all 3 test chemicals. And what we did is we did an inverse prediction to figure out how substantial would the reduction in length need to be to get us to 80% mortality or 95% mortality in our population. So the yellow represents the snout vent length the relative snout vent length that would need to be achieved in order to get to 80% mortality. The red indicates the relative snout vent length that would be needed to get to 95% mortality. Essentially, if you look at that yellow line, what this is saying is that to get to an 80% mortality, the 5-day post-fertilization length would need to be reduced by 14%. To put this into context for you, I've shown the growth data from the cadmium exposures, and a 14% reduction in length relative to the controls is shown by that yellow line. So what you'll see is, particularly in that 80 microgram per liter group, we're going to have a large portion of fish in that group that have lengths lower than that threshold, that 14% reduction threshold. So because of that, what we did here is we actually used snout vent length as an endpoint to estimate mortality. What I mean by that is any fish at the end of the FET test that had a length that was had a relative length of 0.86 or less was counted as a mortality for the purposes of LC50 generation. So I want to show you how the inclusion of that length metric influences FET test LC50 values. So here what you're seeing is the LC50 value for cadmium generated in for 3 independent test replicates. And this is just assuming the standard FET test endpoints of coagulation, lack of heartbeat, lack of somite development, and lack of tail detachment. So when we're just using the standard FET test endpoints, the LC50 value for cadmium is 8,285 micrograms per liter. However, there we go. However, when we include snout vent length, and when we say that larvae, in this case for cadmium, that 14% reduction is equivalent to a 2.99 millimeter snout vent length. So when we assumed that any larvae shorter than that would die, basically what we found is that the LC50 value dropped all the way down to 108 micrograms per liter. So a pretty big difference between these 2 FET test values. So just to put this into a little bit of additional context, when we compare the FET test LC50 value that includes snout vent length as a marker of mortality we get an LC50 value that is quite close to the LC50 value for cadmium generated through the LGS test. So when we're thinking about the sensitivity of the FET test relative to the LGS test, what we've shown here is that if we add snout-vent length as a marker of mortality, we can bring the FET test closer in line to the LGS test. The next endpoint I want to talk about is pericardial area or pericardial edema. And here what you're seeing is the results of our analysis of pericardial area in response to cadmium. So this is pericardial area measured at the end of the FET test, and you'll notice that we have a really nice dose response curve in which increasing concentrations of cadmium lead to increases in relative pericardial area. Again, just for visualization purposes, I have shown on the right-hand side here what these larvae or eleutheroembryos at the end of the FET test may look like. So here we've got this increase in pericardial area, and of course we wanted to link that increase in pericardial area to one of our ecologically relevant endpoints. Because swim performance is an indicator of cardiovascular performance, it made sense to see if we could link those two, so that's the data that I'm going to show here. And what you'll see is when we measured swim performance, and for those of you that are familiar with swim performance, generally when we assess swim performance, we usually use a metric called UCRIT or critical swimming speed, so that's how we measure swim performance in our case. And what you'll see is that we only have data for 3 of our exposure groups, and there are no differences between those 3 exposure groups. We do not have data for the 40 and the 80 microgram per liter cadmium groups, and that's because those individuals did not survive. So it's a little foreshadowing for what you're going to see next with regards to delayed mortality. And when we tried to correlate relative pericardial area with UCRIT values, we saw no relationship between the two. So pericardial area, at least in our case, was not a good indicator of changes in swim performance at 14 days post-fertilization. So of course, the next thing we wanted to do was to look closer at delayed mortality. So again, here we did an inverse prediction using data from DCA, PFOS, and cadmium. And in this case, really what we're trying to do is figure out how severe does the pericardial edema have to be in order to induce 80 or 95% mortality. So the 80% mortality threshold is shown by the yellow line, and then the 95% mortality threshold is shown by the red line. And essentially what we found here is that to achieve an 80% mortality, relative pericardial area would need to be 3.54 or greater. And when we say relative pericardial area, we mean relative to the control group. So the pericardial area would need to be 3.54 times larger in the exposed animal relative to the control group in order for us to achieve 80% mortality. When we look at our data, we see pericardial areas that exceed that threshold quite frequently. So here in the bottom bottom right-hand corner, what you're seeing represented by that yellow line is that 3.54 threshold. And you'll see that we're going to have a large number of individuals, particularly in that 80 microgram per liter group, that exceed that threshold. And we're also going to have some individuals in that 40 microgram per liter group that exceed that threshold. Because of that, we did decide to use this threshold as a marker of mortality for the generation of LC50 values. So here you are seeing the LC50 values for a standard typical FET test, right? So again, it's that 8,285 microgram per liter LC50 value. And again, that's only taking into account the 4 traditional endpoints for the FET test. When we add pericardial area, and when we assume that any fish who has a pericardial area greater than 3.54, if we count those fish as dead and then recalculate the LC50 value, it drops our LC50 value all the way down to 56 micrograms per liter. And if we are to compare that to our LGS test LC50 values, you'll notice that it gets us much more in line with the LC50 value for the LGS test. So in this case, as was the case with growth, we see that the addition of pericardial area as a FET test endpoint really improves FET test sensitivity, bringing it more in line with the LGS test. So to conclude, essentially what we found here is that of these three developmental abnormalities that we were interested in, snout vent length and pericardial area seemed to offer the most promise as additional FET test endpoints. One of the things that you may have noticed as I was going throughout the talk is that I didn't really talk about all of the ecologically relevant endpoints that we measured. So you didn't see mention of sea start. You saw only mention of swim performance once, only mention of feeding behavior once. And that's really because we didn't always see a lot of changes in those endpoints. The one endpoint that we were able to really see changes in and connect back to these abnormalities was delayed mortality. Excuse me. So here we think that snout vent length and pericardial area could be really nice FET test endpoints. And sort of going back to this debate that really stimulated a lot of this work, I think in some ways, both groups are right. Excuse me. We definitely show evidence that indicates that if an abnormality is severe enough, the larvae or eleutheroembryo is going to die. But if this abnormality isn't so severe, it may be that they can recover. So in this case, I suppose everybody is right. But what we would propose is that snout vent length and pericardial edema be considered as additional endpoints for LC50 estimation in the FET test, because they do seem to improve performance. One of the challenges that I think comes with the inclusion of snout vent length and pericardial edema is that you have to actually measure it, right? So we actually have to go in, we take pictures, and then we actually assess the severity using a quantitative metric. So we're using image data to quantify these things. In the ideal world, we would not have to quantify this stuff. We would just be able to look and say, oh yeah, that's really bad. That embryo is going to die. Or it's not so bad. We can count that guy as a survivor. So one of the things that I'd really like to see happen moving forward is the development of an abnormality severity index that allows us to move away from having to measure these endpoints, and that can also account for multiple abnormalities. As I was showing you images, hopefully you recognize that these abnormalities don't occur in isolation from one another. Usually we see embryos with multiple abnormalities, right? So this guy that you're seeing on the right here, he's got a small eye, he's probably shorter than all of his friends, he definitely has pericardial edema, it looks like he has some craniofacial malformations, something's going on with his somite development. So this little guy has all sorts of things going on, and it would be great if we could come up with some sort of severity index for those abnormalities that we could use as a more holistic FET test endpoint. So before I take questions, I do want to acknowledge all of the students that have worked on this project, particularly Julie. Julie Schickfa was really instrumental in this work. Almost all of the data that I've shown you today, Julie was responsible for collecting and analyzing. She built our swim chamber, she built our sea start arena. Super, super talented. This work would not have happened without her. And I can also say that a lot of the images that were included in the PowerPoint today were actually developed by Julie. She's a pretty fantastic graphical design artist as well. In addition to Julie, Kyle Rausch, Dean Stevens, Sarah King, Abby Hawkins, Asal Saeed, Gabby Lamentier, and Alexis Olivas also participated in this research. And I also should thank the TCU Machine Shop, Gary Chizinska, who's our electrician, my husband, Eric Jeffries, and then the TCU Fab Lab, because all of these individuals helped build all of the specialized equipment that we had to use to assess some of these ecological endpoints. In addition, I do want to thank the funders of the research, the American Association of Laboratory Animal Sciences, and then Julie actually received a dissertation grant from CTAC and Procter & Gamble that really supported a lot of this work. And of course, some of the initial work that we did was supported by HESI, and TCU has been incredibly supportive of this work as well. So with that, I will take any questions that anyone has. And if we aren't able to get to all the questions today, I provided my email up here. And then I would also encourage you to check out the paper that resulted from this work. So I put just a little screenshot of the paper there, as well as how you might be able to track it down. So I am happy to take any questions that anyone may have.
Thanks so much, Marlo. That was really wonderful. I'll kick off the questions. We do have a few in the chat that I'll then turn to. But so for the incorporation of these sublethal endpoints, do you think the FET could be utilized more, either as a way to screen or a replacement for the LGS?
I've always, going into this, my hope has always been that we might be able to actually replace the larval growth and survival test with the FET test. I don't think we're there quite yet, and I would not at this point say that the inclusion of these endpoints will allow us to do that. I think we've got a lot more work to do before we get there. And then I also, one of the things with the fish embryo toxicity test, particularly with fathead minnows, is there are some challenges with fathead minnows. So in Europe, they're using zebrafish. Zebrafish have this amazing ability to just spawn on cue. Fathead minnows don't really do that. If you look through the literature, every little thing now, they spawn the first 4 hours that the light comes on. No, they don't. They'll spawn at 2 o'clock in the afternoon. They'll spawn at 11 p.m. I'm pretty sure the lights can be out, and they'll spawn. We find eggs all the time at hours that we would not predict. So while I think the fathead minnow fish embryo test does have some promise, it also has some challenges associated with it that I'm not quite sure we've overcome yet. In my lab, we've definitely done some things to sort of optimize breeding so that we are able to sort of collect eggs in a more synchronous pattern. And then obviously the inclusion of these endpoints helps get us closer to LGS LC50 values. But I do think there's quite a bit more work that needs to happen before I would feel comfortable saying it should be a replacement, but that's definitely what we're aiming for, is to get to the point where it could be a replacement.
Thanks. So start going through the questions in the chat. So Marco Dilger asked, what are the disadvantages of doing the FET test without the chorion? Would the LC50s be expected to be lower?
Yeah, so this question actually comes up a lot when I talk about the FET test and whether or not we should dechorionate these embryos. And I would say probably every 3 years in my lab, we have a discussion about whether or not we should try these tests with dechorionated embryos. And the reason that we haven't is really because dechorionating the embryos, you can either do it chemically or you can do it mechanically. I think as a toxicologist, anytime I'm adding a chemical to a test, I always get a little nervous, right? Because you think, well, wow, could this chemical like impact toxicity of chemicals that I'm actually going to expose the fish to? So we've always tried to avoid any kind of unnecessary chemical exposures. And then the mechanical dechorionation, the amount of time it would take would just be extraordinary. So I think if we're using the FET test in the context of regulatory toxicology, and if it's going to be something that's done by contract labs, I think it has to be simple and it has to be straightforward and it has to be easy, because the LGS tests are super easy. We can get an LGS test up and running in probably an hour. If you were to dechorionate embryos, I do think it would, for some chemicals, definitely improve the performance of the FET test. I just don't think it is feasible in terms of timing. So that's why we've not chosen to do it. There have definitely been people that have looked at dechorionation and they do find that for some chemicals it does enhance performance or make it more similar to the FET test, or sorry, to larval growth and survival tests or to adult or juvenile tests.
Yeah, thanks for that, Marlo. I know in the zebrafish world, it's been a debate as well, and some labs do it, some labs don't. So I know there's been a lot of work to compare different performances in zebrafish embryo labs. So I wonder if some of that could maybe give an answer ways. But like you said, you have to do something either chemically or mechanically to them.
Yeah.
All right, next question. Any speculation on why the FET test is less sensitive than the LGS? Could it be related to bioavailability with embryos more protected from exposure? And that's from John Allran.
Yeah, I think that's definitely an option. And we do know there's actually been some really nice work that has, particularly in zebrafish. I'm not aware of anyone doing this in fathead minnows, but there's been some really nice work looking at what size compounds can actually cross through the chorion of zebrafish embryos. And there definitely is a size limitation. So if we're dealing with large molecular weight compounds, they're going to have a much more difficult time passing through those pores in the chorion. And if the chemical is not bioavailable to the embryo, if it's getting bound up in the chorion or it's not even able to access the chorion at all, then you're basically just not getting an exposure of the developing embryo. So that definitely is one reason why we see less sensitivity in the FET test. Another reason that some have hypothesized with regard to reduced sensitivity is just metabolic capacity of embryos versus larvae. So during early periods of development, the embryos may not be able to biotransform some chemicals into sort of bioactive, more toxic metabolites. So that's one hypothesis. And then it does seem like for neurotoxicants, we do see reduced sensitivity. And that may just be related to the timing of key developmental events related to neuronal function.
Great, thanks, Marlo. I think this one maybe somewhat goes along with your last answer, but are some of these sublethal endpoints specific to mechanisms of action? For example, can we predict which types of chemistries may not be well suited? And that's from Adele Strobel.
So one of the things, so I think we could definitely identify endpoints that are specific to certain modes of action. What we in my lab, what we've really tried to do here is identify endpoints that seem to be more general, right? So pericardial edema, we see it in response to just about everything. The mechanism of action of different chemicals that leads to the pericardial edema is likely very different across chemical classes, but we see it across multiple classes of chemicals. Same thing with eye size, same thing with growth. So the endpoints that we've picked, we've really selected to sort of represent endpoints that are common and not necessarily tied to a specific mode of action. I can tell you when we were doing gene expression work, those were a lot more specific, right? So for example, with gene expression, if we were looking at biomarkers of oxidative stress, we were thinking of things like cadmium, right? Chemicals that we know induce oxidative stress. Yeah, so we've sort of focused on the more general stuff because we want whatever we do to sort of be broadly applicable.
Thanks, Marlo. And do you have a hard stop at the top of the hour, or can you take a few more questions?
No, I can take a few more questions.
All right. Adele also asked, how did you pick your dose ranges? How would you pick an appropriate dose range?
So for this study, I will tell you, we actually had a heck of a time picking our dose ranges. And the reason that it was difficult is because the sensitivity of the embryos is so different from the sensitivity of the larvae. And so what we did find is when Julie had tried to do some of these tests with concentrations that we would normally use if we were just running a FET test. A lot of times she would actually see that 2 days later, everybody in the 2 highest doses would be dead. So for this particular study, we did have to sort of decrease our concentrations a little bit just so we didn't have such high mortality at like day 6. So the dose response range is a little difficult here. In general, what we do when we're trying to figure out concentrations for FET tests, LGS tests, we usually just conduct range finding tests or limit tests to say, okay, well, when do we get 100% mortality? When do we get 50% mortality? And then we sort of select concentrations from there.
Sorry, unmuting. Thank you.
No, you're fine.
Stefan Schultz had a question, but I think we answered it. Stefan, if you feel we didn't write in. So then the next one from Mark Jankowski, he said, apologies if he missed the part, but how long after the treatment was removed was the mortality observed?
So we removed the exposures at 5 days post-fertilization, which is the end of the FET test, and then we grew out the larvae in clean water for 9 days.
And then Mark asked, was the relationship between the snout, vent length, pericardioedema, and latent mortality quantitative? And that's yes, right?
Yes, yeah.
All right, again from Stefan Schultz for the FET, have you looked at the swim bladder inflation?
We have not, and I think that's actually a really interesting point. So I do a lot of endocrine disruption work as well, and for anybody that works with thyroid disruptors, there's some pretty clear evidence, particularly for thyroid disruptors, that swim bladder inflation could be a really, really nice sort of biomarker, and we have really nice adverse outcome pathways associated with swim bladder inflation as well. It is not something that we've looked at, but it may be worth looking at in the future.
Great, thank you. Edward Lay asked, did cadmium actually get inside the embryos?
So cadmium is an interesting story. So we do think that the cadmium is getting inside of the embryos, but we also have evidence that not all of it is getting inside the embryos. So there is some really nice literature showing that cadmium tends to sort of get bound up in the chorion. So it sort of gets stuck as it tries to pass through. But given that we are able to induce mortality, given that we are able to see some adverse effects, we're confident that to some degree that cadmium is getting in. But in the case of cadmium, that may actually explain why the LGS is more sensitive when we're just looking at the standard endpoints.
Thanks. And Claudia DeMartagazzi had a similar question. Did you measure embryo uptake, and was there a difference between the FET and the LGS?
We did not measure it, but I would suspect that there would be, right? I think one of the things, these embryos and larvae have some fundamental physiological differences, right? So clearly they have the chorion, which is a structural difference, and that's going to sort of limit uptake. But then, right, fish larvae, after a few days, they really start to rely on their gills, and the gills are a really important route of uptake for some of these chemicals. So, so in general, I would guess, and I do not have data to support this, but my educated guess that would be that if you were to measure concentrations of these chemicals in the bodies of the embryos and larvae, we would expect to probably see higher concentrations in the larvae. Assuming that you throw the chorion out of the sort of egg and you're only focused on the actual embryo itself.
Thank you. So Ming Hong has a statement that she asked if you agree with. So they wrote, in terms of overall ecotoxicology assessment, whether to use a FET test or the larval testing still depends on the fish development stage the fish gets exposed to the test substance and how the fish gets exposed to in nature. So we can't completely replace the larval test. Do you agree?
Yeah, I would agree with that. I mean, I think I think the FET test has its place, but I think whenever you're using any toxicity test, you have to recognize the limitations of that test. So yeah, I agree with that statement, is that we have to understand the context in which we're using these tests. So if we're looking to evaluate a test chemical that maybe is only episodically released in the middle of winter when there aren't going to be embryos, we probably shouldn't be using embryos to evaluate the toxicity of that chemical.
Thanks. Yeah, I think applicability domain is really important, and to think, you know, if you get an answer from a FET test, does it mean you do or do not? I think that's something as a field we would need to think, like, how is this used? What's the context? Can it be, you know, a screen? Can it be something when, like you said, like when is it an appropriate replacement is really need to be thought about in the problem formulation.
Absolutely.
And then Ming also had a comment on the chorion, but I think we covered. So then Stefan Schultz, and we are getting near the bottom, so asked for the organic compound she tested, how do the effect concentrations relate to the baseline toxicity? So for example, the predicted toxicity based on the hydrophobicity.
So we didn't really look at those relationships. It would be interesting to go back and look at that, right, to see, can we, you know, sort of look at log KOW values in relationship to the data that we're generating, but that is not something that we have done.
Yeah, great. So that was actually the last question I see. I appreciate the engagement from the audience. I know a few people had to drop off. at the hour. I think this is really promising. Marlo, any concluding comments from your end?
Oh, wow. No, I think, stay tuned. I will say, so one of the things my lab is working on right now is we're actually working on the development of marine FET tests using sheepshead minnows and inland silversides. So we're waiting on a little bit of analysis, chemical analysis, so we can put a percent recovery in a paper that we've written. So hopefully that paper will come out soon. So be on the lookout for that. And by all means, feel free to email me if you've got any questions.
Great. Thank you very much. And thanks everyone for your attention. Have a great rest of your day. All right. Bye, everyone.